to introduce our first speaker, who I've known for many, many years, probably more than 2 decades, Dr. Silvère Van der Maarel from Leiden, the Netherlands. It's an honor and pleasure to introduce Dr. Van der Maarel as our first speaker. And thank you, Silvère. Thank you. And I think they'll need to know how to use the cue. So. Yeah, thank you, Silvère.
Thank you, Dan.
You know it is, all right. Next slide.
Back slide. Okay, thank you, Dan, and thank you, Society, for inviting me to this biannual meeting. It's always a pleasure to be here. It's really great to see so many people who have joined us today. So I'm based at the Leiden University Medical Center in the Netherlands. And since the late 70s, early 80s, our institute has been involved in identifying the cause for FSHD. But it started by 2 gentlemen in the hospital, George Padberg, a neurologist, and Rune Frans, a geneticist, who at that time realized that FSHD is a familial disease and it has a genetic component to it, and we really wanted to understand what that genetic component was. Since then, we have always been involved in FSHD research, and we have really become expertise, or created expertise in the genetic research mainly. And we're doing the DNA diagnosis for FSHD in the Netherlands, and this is accounting for about 400 to 500 diagnostics services each year. We also provide assistance to the DNA diagnosis of those cases where the conventional genetic tests do not confirm or are inconclusive and cannot confirm the clinical observation of FSHD. And we do that for about 250 cases annually on a research basis from really all over the world. So the research that we are doing, we're not doing in isolation in our lab. Modern science, I always say, is teamwork, and there are many, many people all over the world who are contributing to the story that I'm going to tell you today. And my main collaborators, but certainly not all, because I really would like to acknowledge everyone in the field who's really facilitating the progress that we're making right now. But my closest collaborators are listed on this slide. It's Stephen Tapscott and Dan Miller in Seattle, Ravi Tawil in Rochester, and of course also George Padberg and Basil van Engelen in Nijmegen, as well as the others that are listed on this slide. You can also see the people from my lab who are working every day hard to find a solution, a cure for this disease. And I also would like to acknowledge the many different funding that we received from different organizations. I would like to emphasize the importance of societies like the FSH Society, who really allowed us to really stay on the edge of what we know about this disease. It's really societies like these that allow us to perform cutting-edge research, which in hindsight very often is regarded as really what was important and really created the breakthroughs. So since I'm the first speaker today, I took the liberty to introduce you quickly again to the basics of biology and genetics, just to set the stage and to explain you a little bit where we are with FSHD. So for most families, FSHD is an autosomal dominant disorder with incomplete penetrance. For me as a geneticist, this makes sense, but I can understand that for many of you, this might be actually not something that you see every day. So let me try to explain this. So all the genetic information that we have and that we transmit or transfer to our children is located in our DNA. And the DNA is divided over 23 pairs of chromosomes. And these pairs are almost identical, at least 22 of them. The last pair, these are the sex chromosomes, and they differ between male and females. So the 22 identical pairs we call autosomes. So this means that the genetic defect in FSHD is located on one of the autosomes, and that's chromosome 4. As I said, we have 2 pairs of each chromosome. That means that we have 2 times almost identical information. And this information we transmit to our children, and we only transmit one of the 2 chromosomes. So that makes sense because we have 2 pairs. The father and the mother each them transmit one of the two, so the kid ends up again with a pair of a chromosome. Now, sometimes there are little mistakes in the recipes, which we call the genes, the recipes for everything that we are, that build the building blocks of our cells. And sometimes in these genes there are small mistakes. Well, if there is a mistake in one of the two copies and it leads to a disease, then we call it a dominant disease. If there are mistakes in both copies and then only it causes a disease, then we call it a recessive disease. So FSHD is a dominant disease, meaning that only that a mistake in one of the two copies is already sufficient to create the disease. Now the last term, incomplete penetrance. I think you know better than me that in many families with FSHD, we are aware of individuals who do have the genetic defect, but are not presenting with the disease. So this is what we call incomplete penetrance. These are the ones that remain asymptomatic throughout their life. Well, they do have the genetic defect. So the central dogma in the biology is that we are built up of cells, and cells are basically, it's a bag of soup, which we call cytoplasm, and it's surrounded by a cell membrane, and there is an other body within that cell that's called the nucleus. And the DNA is located within the nucleus. And there are two important processes that you need to remind. One is that the DNA needs to be able to copy itself, otherwise we cannot make a daughter cell. And this process we call replication. Sometimes little mistakes are being made during this replication, during this the copying of the DNA, and this can create a new disease. So these are typically the new cases in a family. When there's no family history of the disease and suddenly there is a new case, then most often the genetic defect is caused by this process. But also these recipes, these genes need to be read and need to be produced. And that's a two-step process. First, we transcribe it. We produce a molecule that is almost identical to DNA, which we call RNA. That is then transported to the cytoplasm where the recipe is being prepared into a protein. So this is actually quite a challenge for a cell, because in each nucleus there's 6.5 feet of DNA. And if you would translate that, that means that there's about half a marathon of DNA, the length of half a marathon, wrapped within a tennis ball. So you can see that's really a huge challenge, and it also tells you that it needs to be done in a very structured way. I always like to make this comparison with this spring that I'm showing on this slide, that is also ordered in a very structured way. And please think about our DNA as being ordered in this way. So we have about 25,000 recipes, but we certainly do not want to cook all of them at the same time in the same tissue in our same cell. So basically the way the cell deals with this is that many of the recipes that we don't need, we put away there in this condensed part of the spring. They're off, and this is what we call heterochromatin. At least the DNA is very tightly packed. It's inaccessible, and it really allows us to pack this 6.5 feet of DNA within the nucleus. But some of the recipes we need to be able to read and to prepare, so that we put in this open part of the spring, where it's accessible. We can read it and we can produce it. So that's the open part, and on the left side of this slide you can see the way that we try to show this in a very elaborate way. But please, again, look at this spring. This is how we organize our DNA. We have open structures and we have closed structures. Now, in 2010, we published a paper where we said that FSHD is caused by inappropriate production of the DUX4 protein and muscle of FSHD. And this was really the work of many people around the world that really culminated in this one particular paper. And we thought that this was really regarded as a breakthrough. And I thought what might be nice today is really to reflect on that and see what has happened since then, and was this really a game changer in the field. And again, I would like to emphasize that this is not the work only done by my lab, but is really the product of many researchers, also that I'm seeing here today in the audience, that really all together and finally produced this uniform mechanism for the disease. So what did we learn? Well, most individuals with FSHD have a contraction of a repetitive structure in chromosome 4. And I'm showing this with these triangles. So in normal, healthy, controlled individuals, there are about 11 to 100 of these copies in an array. And this allows it to be in this closed part of the spring, as I'm showing here with these orange circles. In most individuals with FSHD, we see that there's a contraction, leaving only 1 to 10 of these repeat units. And the consequence of that is that actually it moves to this more open side of the spring, so it becomes accessible. And if there are any recipes in there, suddenly these recipes can be prepared in the muscle, where they're normally not prepared. And this really leads to the production of this DUX4 protein. And you really don't want that DUX4 protein in your muscle. And again, studies by many people, Stephen Tapscott, Alexandra Bellew, Dan Miller, Scott Harper, and many others have shown the deleterious effects of DUX4 in skeletal muscle. So this is not produced in normal healthy individuals, but becomes produced suddenly in individuals with FSHD. Now there's also a small group of patients where we also saw that this repeat structure moved into this open part of the spring, but there was no contraction. And for a long time, we really didn't understand these individuals. So the primary disease mechanism, once again, is that normally we have a long array. It's there up in this closed part of the spring. And in FSHD1, because of this contraction, it moves to this open part of the spring, and suddenly this DUX4 protein can be produced in muscle. Now, almost 2 years ago, we actually realized that in the individual who's FSHD2, so there is this opening of the chromatin structure, or of the structure of the repeat, but there is no contraction, that these individuals carry a different genetic mutation, different genetic lesion. That's a mutation in a protein that's called SMCHD1. And by now we realize that about 85% of individuals with this condition really have a mutation in SMCHD1. So what is SMCHD1? Well, SMCHD1 is a protein that binds to this repeat structure, and it is necessary to keep it in this closed environment in the spring. So if you have a mutation, you basically have reduced activity of this protein at this repeat structure, again, causing it to move to this open part of the spring and the production of DUX4. So the important news here is that these two forms of the disease very quickly converge at the same mechanism. It is opening up of the repeat structure and the production of this DUX4 protein. There are actually other proteins that also are involved in keeping this repeat in this closed part of the spring. We are pretty confident that there must be other FSHD2 genes out there, because we cannot explain all of our families with FSHD2. But there is also a very different condition, which is in primary immune deficiency, that also that has mutations in other genes that are also involved in the structure of this D4C4 repeat. And we like to use to study these diseases as well, because it gives us the basic information of what is minimally necessary to keep this repeat in this closed part of the spring, and that may actually lead to new leads for therapy. Now, one thing that I'm particularly interested in is this clinical variability. I already started my talk today about this observation that about 20% of gene carriers, so carriers of this mutation, will never see a neurologist because they remain asymptomatic. And I think that's a very intriguing finding, and we should learn about these individuals, because these individuals can tell us why they are protected from the disease. And I think that's important for us because it will provide us new leads for therapy. So, and for a long time we had no idea what was causing this, and I think we're starting to understand this. And this was really through the work of one of my collaborators, Sabrina Sacconi in Nice, who really recognized these families where you have this typical progression of the disease. You have the onset in the early or the late teens, early 20s, the slow progressive phenotype that we really see. This is the typical FSHD phenotype. But she had these individuals within these families that had a much more severe course of the disease, a much earlier onset, much more severe phenotype. And it was really through this observation that we could realize that in these individuals, we have actually the co-segregation of FSHD1 and FSHD2. So these individuals not only have a contraction of the repeat, but they also have a mutation in SMCHD1. And that really provides us now a very complete picture of what's going on the genetic basis in FSHD. So in normal healthy controls, we have the repeat structure, which is in this closed spring structure. It's not producing DUX4 protein. And that is also partly because of the work that SMCHD1 is doing to keep it there. In FSHD1, we have this contraction, which leads to a relative opening of the spring and the production of DUX4 protein. And then we have FSHD2, where basically the protein is missing that is necessary to keep it in the closed structure. And again, we see this relative opening of the spring structure and the production of DUX4. And then we have FSHD1 and FSHD2, where both genetic flaws come together. And you see a much wider opening of this spring, so it's much more accessible and much more DUX4 is being produced, leading to a more severe phenotype. I think that really the most important thing about this is that we now realize that there are other modifiers in our genome that are important to keep this repeat in a closed structure. And also some recent work that we are now trying to publish is really showing that also in FSHD1 families, so where there is no involvement of FSHD2, we're starting to see the same phenomenon. And the important thing that we are seeing here, again, you see this variability in disease presentation indicated by the color codes, being green are carriers of a mutation but unaffected, red the very severe ones. And again, I've indicated these springs again. And I think the most important thing here is that we now see that those that remain unaffected also have this closed repeat structure. Although they have the contraction, they have this closed repeat structure that is indifferent from controls. So we strongly believe that with the current knowledge of the disease mechanism, we already need to work on treatment. And there are many points that we can start thinking about. We can think, of course, can we prevent the opening of this repeat structure? Or perhaps reverse it? Or can we prevent the production of this RNA or the DUX4 protein? Or can we prevent the action of the DUX4 protein itself? Or maybe one of the downstream targets? So the take-home messages from my side today are that there are at least two forms of FSHD. The common form, which is caused by contraction, and the rare form, most often caused by mutations in SMCHD1. Both forms can be genetically confirmed with great accuracy. Both forms have an identical disease mechanism. It's the expression or the misexpression of DUX4 protein in skeletal muscle. Some individuals have FSHD1 and FSHD2, and this at least provides a partial explanation to the variability that we see in this disease. I think that we really have uncovered the mechanistic basis of this disease. And it is really through the work and the help of all of you, providing every time new samples to us to allow us to make this progress. So thank you very much for your attention.